Today we're going to talk about lab technique and media. In the lab, prokaryotes are isolated and grown in pure culture in order to identify them and study their growth patterns. A pure culture is a population of only one bacterial species. Pure cultures are obtained using special techniques. All materials used must be sterile or free of microbes before usage. Then they are handled using aseptic techniques, which are procedures that minimize the chance of other organisms being accidentally introduced. The culture medium is the medium that the cells are grown in or on, is a mixture of nutrients dissolved in water and may be a liquid broth or a solidified gel-like form, kind of like Jell-O. So if you see this picture here, this is a pure culture because it's only one species, and this is a mixed culture because you can see there's several different species. This is an agar plate. So what you have here is a petri dish that you fill with agar. Agar is the nutrient medium that you're going to be growing their bacterial culture in. Cultivating bacteria on a solid culture medium, and specifically agar plates. This requires the solid culture medium, a petri dish, and an inoculating loop. A single bacterium supplied with the right nutrients will multiply on the medium in a limited area to form a colony, which is a mass of cells all descended from the original one. About 1 million cells are required for a colony to be easily seen with the naked eye. Agar, which is a polysaccharide extracted from marine algae, is used to solidify a liquid culture medium. It is first sterilized by heating it, which liquefies it, and then it hardens as it cools to form a gel-like medium. The culture medium is contained in a petri dish, a 2-part container made of plastic or glass. It excludes airborne microbial contaminants. A petri dish containing a medium is referred to as an agar plate. So this diagram is showing you the streak plate method. It's the simplest and most used technique for isolating bacteria. So first, you sterilize your inoculating loop. So this little guy right here, it has a handle. Sometimes it's wood, sometimes it's plastic. And then this long piece is a wire, so it's metal, so it can conduct heat, okay? And it's got a loop on the end, which is why it's called an inoculating loop. So first you have to sterilize your loop in the Bunsen burner, so in the open flame. You hold it in there until it turns bright red, and then you pull it out and let it cool off for a minute before you put it into the culture, because you don't want to kill, and scorch the bacteria cells. So then once it cools off, you dip it into the culture. So you're basically getting some of the bacteria on the loop. Then you lightly draw several times across an agar plate. So you can see right here, you make your first set of streaks, okay? So that is the bacterial cells that you just pulled out of that tube. And now you go sterilize your loop again. So you hold it in the flame for a few seconds till it turns bright red. Then you pull it out of the flame, let it cool off for a minute. And then you're going to make your second set of streaks. So your new set of streaks are made at an angle to the first set. And so what you do is you touch your first set, and then you make your streaks in the second quadrant here. Then you sterilize your loop again. So you hold it in the flame till it turns bright red, pull it out, give it a second, let it cool off. And then you're going to make another set of streaks from your second set of streaks. So you go from the second set here, you start here, and you drag your loop across to make this set of streaks. And then you go and you sterilize the loop again, because you're not going to use it anymore now, but it needs to be sterilized. So the last set of streaks, you can see you're getting less and less concentrated, right, of the cells. By the third set of streaks, the cells should be separated enough so that the isolated colonies can develop after you put them in an incubator. So you can see these little dots here are going to be the beginning of your colonies that are growing from this streaking method after you incubate it. Typically, depending on the species, It might be 24 hours, it might be, 48 hours. It all depends on the species that you're using. We're going to watch some of this video together real quick.
Hello there. First video, we're going to show you some of the basic aseptic techniques that we use in microbiology. You'll get to practice these plenty in class, but we want to give you a little video that shows you the basic techniques. First thing I'm going to show you how to do is to use the inoculating loop, which is a very typical tool we use to move bacteria from either a culture onto a plate, from a culture tube to another culture tube, from a plate to a plate. Now, there's the transfer of bacteria from one area to the other. Most important thing is when you're using the loop, it needs to be sterilized. There's a proper technique for sterilizing a loop, which I'll show you right now. With the Bunsen burner going, we're going to heat the loop starting up closer to the handle and slowly moving our way down to the end so that if there was any liquid here, it would first evaporate and then burn off any of the bacteria versus aerosolize and potentially spread any of the bugs into the air. So put the loop in the flame, you'll see it will get red hot. Slowly move it down the flame, which will kill off anything that's on it. And eventually ending with the loop, which will get red hot, particularly the first time you use it during the day. You don't know where it's been. And once it is sterilized, you pull it out of the loop. Now we need to let it cool off. Things to not do when you cool it off is don't blow on it. That won't help matters. Don't stick it into a beaker of cool water because the water won't be sterile. Basically, you have to let it cool off in the air, or we'll show you a technique later where you can touch it to a sterile area on a plate or on condensation in the plate if you're in a rush. Okay, so what I'm going to do is first I'm going to move some bacteria from this culture I have that I started yesterday of E. coli and do what is known as a streak plate onto this plate. Before we get started, though, there's some important record-keeping information we need to talk about. Namely, on every tube or on every plate, you need to record 4 important pieces of information about what's going to end up on the plate. Before you put anything in, you need to label the name of the organism, the type of media, in this case Na, which stands for nutrient agar, initials of who did the work, the spreading, SDM, that's me, and the date at which the inoculation was made. Trying to keep it around the edge of the plate so that anything that's on the plate will be easier to see once they grow. Same kind of thing is done on a label. 4 corners, the bacterium, the date, the investigator or the student, whoever it happens to be, and the type of media. And if it's nutrient agar or nutrient broth, I'm stating up all those pieces of information. Important to make sure you get those all on there. So I'm going to move the bacteria that I have growing an overnight culture of E. coli. You can see in this culture tube, which has these lids on it that slide up and down. They don't screw on, which is going to be important because there is a technique which allows us to open and close these with minimal chance for contamination. And move it on to the plate, which most of you are familiar with, a Petri dish that has a lid on the top and has nutrient agar on the bottom. It's stored inverted, and then we'll talk about that as we show you how to do that. So if you're right-handed, having an inoculating loop in your hand like this, not sort of grabbing it like this. It'll be a lot easier if you get used to holding it sort of like a pencil, particularly so you have these 2 pinkies available to open and close tubes, as you'll see in a moment. So first thing I'm going to want to do is sterilize the loop. Again, put in the flame, slowly bringing it down, and I'm going to get hot. Now that's sterilized. It's cooling. Talk about how to open and close the lids. Since I'm right-handed, I'm going to use my pinky of the hand that has the inoculating loop literally to grab the lid. Thing you want to get used to. Every time you open it, open, flame, flame, close. By flaming it, I'll show you what I say. I'll grab the lid, if I pull it straight off and then just run it through the flame, I can do the work I need to do, flame, and close the lid. The flaming heats the air above the liquid, which causes it to expand and push out, which minimizes the chance for contamination. Never want to set the lids down. You always have them in your hand so that these have all been through the autoclave and everything started sterile, so you want to minimize potential contamination. So my loop is cooled now. What I'm going to do is flame open, go in. It'll pick up a little loop of liquid, which you probably can't see. Flame closed. Now I can set this down if I want, since I'm going to be doing a streak plate. I have a little area on this divided into 3 sections. What I'm going to do is first flip the plate over and then spread starting at the top back and forth to get down here to the middle, and then I'll sterilize the loop, and you see drag into it a little bit and try to spread it out. We're basically trying to spread the liquid out and eventually get isolated colonies. Again, we don't want to take the lids off of these ever. We want to tip it, and basically flip and tip. And we'll flip it, tip it, get in there, streak it, and then put it back down. So I'll flip it over, and tip it up, get in, let the loop run gently on the surface all the way to the middle here, close. Now I'm going to re-sterilize this, and once you've used it and you know there's nothing up on the shaft of it, you can sort of go in quicker, get the loop. I'm going to turn the plate now 90 degrees, and I'm going to dip once into where I had left off before, and then streak into this quadrant, and then sterilize and do the same thing again. Again, just tipping it, go in here into where I had left off, go into here and spread it out, re-sterilize the loop. I'll turn the plate again 90 degrees, and one last time I'm going to try to dip into the area where I just streaked and drag it out and dilute it even further into the last quadrant. Tip, go in here, streak it out, done. When you're done, sterilize the loop. This could now be set down and flip the plate. Plates always get flipped, stored this way, minimizes the chance of anything falling on the surface and contaminating it. So there is a way to move liquid from the culture to the plate.
So we're going to talk about the different types of media that can be used to grow different species of bacterial cells. So complex media contains a variety of ingredients like meat juices and digested proteins that act as a tasty soup for the microbes. You can have blood agar, which is more rich than nutrient agar and includes the nutrient hemin that's found in blood. You can have chocolate agar, which contains lysed red blood cells and additional nutrients. It's chocolate in color, not in taste. Chemically defined media is composed of precise amounts of pure chemicals. This is essential when studying nutritional requirements of bacteria. Selective media inhibits the growth of organisms other than the one being grown. This can be complex or chemically defined. So you're selecting for the one organism that you want to grow. So it's not going to be conducive to the other organisms or other strains of the species. Differential media contains a substance that certain bacteria change in a recognizable way. This can be complex or chemically defined. So you're differentiating between the bacteria, remember, like gram-positive or gram-negative. Enrichment culture is an enhance the growth of one particular species in a mixed population. Broth medium is a nutrient-infused liquid media used for growing bacteria. Nutrient broth is typically made of a powdered beef extract that contains peptones, which are broken down proteins. The powder is dissolved in water, put into test tubes, and then sterilized by heating it up. Broth is convenient as most bacterial samples can be easily introduced into and grown in this type of medium, even those with widely different aerotolerance, remember their oxygen requirements. Aseptic technique is a procedure done under sterile conditions, which means that it's free of all microbes. This is performed by doctors, surgeons, nurses, microbiologists, laboratory workers, etc. The goals of aseptic technique are to prevent contamination of specimens and to prevent contraction of diseases that the person is working with. So let's look at the picture of this scientist here. What kind of aseptic techniques do you think he's using? So to prevent contamination, he's wearing gloves, he's wearing goggles, he's wearing a lab coat. He's working in a hood, so you can see that glass that's between him and the sample that he's working with. He's not touching the materials, right, with his bare hands. He's not breathing on them. So he's trying to prevent contamination of the samples while he's working with them. To inoculate a broth culture, hold a labeled broth tube in your left hand and your loop in your right hand using the thumb and the first two fingers. Flame sterilize the entire loop length and then let it cool. Remove the cap from the broth tube with the little finger of your right hand that's curling against your palm. Flame the opening of the broth tube, touch the loop to the desired colony on the plate, and then stir the loop around in the broth. Reflame the opening of the tube, return the cap, and place the tube in a test tube rack to be incubated. Flame sterilize the loop again. So you would do this procedure if you're taking samples of a bacterial culture from a plate and you want to grow it in a broth tube. Bacteria grow tremendously fast when supplied with an abundance of nutrients. Different types of bacteria will produce different looking colonies. Some colonies may be colored, some may be circular, others are irregular in shape. The characteristics of a colony, the shape, the size, the pigmentation, are termed the colony morphology. Colony morphology is a way scientists can identify bacteria. In fact, there's a book called Bergey's Manual of Determinative Bacteriology, and they commonly call it Bergey's Manual, that describes the majority of bacterial species identified by scientists so far. This manual provides descriptions for the colony morphologies of each bacterial species. The form refers to the basic shape of the colony. Is it circular? Is it filamentous? Right? The elevation is what is the cross-sectional shape of the colony. Turn the petri dish on its end. The margin, what is the magnified shape of the edge of the colony? The surface, how does the surface of the colony appear? Is it smooth? Is it glistening? Is it rough? Is it dull? Is it wrinkled? The opacity, is it transparent? Is it clear, opaque, translucent? The chromogenesis or the pigmentation, is it white? Is it buff? Is it red? Is it purple? What color is it? These are some examples of the form, elevation, and margins. For bacterial cells, each distinct circular colony should represent an individual bacterial cell or a group that has divided repeatedly. Being kept in one place, the resulting cells have accumulated to form a visible patch. Most bacterial colonies appear white, cream, or yellow in color and fairly circular in shape. This is an example right here of Bacillus subtilis, which is a bacteria commonly found in the soil. The one on the right here is Proteus vulgaris. It's a pathogen, meaning it causes harm to the host, and commonly causes UTIs, which are urinary tract infections. This one is Staphylococcus aureus. It's normal flora, so it's always found on or in your body, but it can be pathogenic when it has the opportunity to. So when your immune system is down or something happens, it can take over and cause an infection. If you remember, MRSA is methicillin-resistant Staphylococcus aureus, so it's a form of this bacteria that's resistant to an antibiotic that we use to kill it, which means it's a lot harder to get rid of. Streptococcus pyogenes is this one over here, and this is pathogenic. It causes strep throat. And then down here at the bottom, we have some yeast. Yeast colonies generally look similar to bacterial colonies. Some species, such as Candida, can grow as white patches with a glossy surface. So this one over here is Candida albicans. It's a yeast, and it causes yeast infections, and it can be in anybody, women, men, even babies. The middle one is Saccharomyces cerevisiae. This is your baker's yeast. So this is what we use when we make bread. And then the one on the right here is Trichoderma harzianum, which is a green mold. As you can see, they look pretty similar with the naked eye for the most part. So that's where microbiology comes in when we use microscopes, and we'll look at their actual structures, the shapes of their structures, their cluster orientations, and we'll do all of that in the next unit.